அனைவருக்கும் வணக்கம். பயாலஜி தமிழ்நாடு சேனல் சார்பாக உங்களை அன்புடன் வரவேற்கிறோம். இன்றைக்கி நம்ம என்ன பார்க்க போகிறோம் அப்படின்னா, உங்களுடைய ஸ்டேட் போர்டு பயோ பாட்டனி 12th ஸ்டாண்டர்ட் இங்கிலீஷ் மீடியம் புக்கில் இருக்கக்கூடிய பிரின்சிபல்ஸ் ஆஃப் பயோடெக்னாலஜி 4வது பாடம், அதில் இருக்கக்கூடிய 1 வேர்ட்ஸ் கொஸ்டின்ஸ் அண்ட் ஆன்சர்ஸ் பார்த்துட்ருக்குறோம். அதில் பார்ட் 4ல் நாம் சந்திக்கிறோம். நேம் த சப்ஸ்டன்ஸ் கன்வீனியன்ட் ஃபார் செப்பரேட்டிங் DNA ஃப்ராக்மெண்ட்ஸ். Agarose. Mention the size of DNA can be isolated by agarose gel electrophoresis. Few hundred to 20,000 base pairs. Which chemical is useful to separate smaller DNA fragments? Polyacrylamide. DNA molecule is dash charged. Negatively charged. Which technique is performed with marker DNA fragments of known size which allow accurate size determination of an unknown DNA? Agarose gel electrophoresis. In agarose gel electrophoresis, the brands of DNA in the gel are stained with the dye called ethidium bromide. Which color is established when UV light is placed on ethidium bromide? Orange. Name the technique includes variety of tests that are used for detection of pathogens in plant tissues. Agricultural diagnostics. Expand ELISA. Enzyme-linked immunosorbent assay. Which technique is a diagnostic tool for identification of pathogen species by using antibodies and diagnostic agents? ELISA, enzyme-linked immunosorbent assay. Which diagnostic technique is used in plant pathology, especially for weeding out virus-infected plants from large-scale planting? ELISA. DNA probes, isotopic and non-isotopic, are popular tools for identification of viruses and other pathogens. Which techniques are widely used analytical tools for the specific identification of desired DNA or RNA fragments from larger number of molecules? Blotting. The process of immobilization of sample nucleic acids or solid support nitrocellulose or nylon membranes is called blotting. Name the substance used as target in the hybridization experiments for their specific detection. Blotting nucleic acids. In which technique DNA transfer from agarose gels to nitrocellulose membrane? Southern blotting. In which technique the transfer of RNA to nitrocellulose membrane is performed? Northern blotting. In which technique electrophoretic transfer of proteins to nitrocellulose membrane takes place? Western blotting. Southern blotting was introduced by Southern 1975. The transfer of DNA from agarose gel to nitrocellulose filter paper is achieved by capillary action. Name the substance used as buffer in agarose gel electrophoresis. Sodium saline citrate (SSC). In agarose gel electrophoresis, the DNA is hybridized with a nucleic acid and can be detected autoradiography, in which technique that captures the image formed in a photographic emulsion due to emission of light or radioactivity from a labeled component placed together. Autoradiography. Which nucleic acid is not binding to cellulose nitrate? RNA. Who devised a procedure in which RNA bands are transferred from the agarose gel into nitrocellulose filter paper? Alvin et al. Northern blotting, 1979. Which filter paper is used for northern blot? Amino benzoyloxymethyl paper prepared from Wattmann 540 paper. Which technique refers to the electrophoretic transfer of proteins to blotting papers? Western blot. Which filter paper can be used for Western blot technique? Nitrocellulose. Which biomolecule is identified by probing the blot with the radio-labeled antibody which binds on the specific protein to which the antibody was prepared? Protein. Gene targeting experiments have been targeting the nuclei, and this leads to gene knockout. What are the two types of vectors used for gene knockout? Insertion vectors and replacement or transplacement vectors. Name the vector that are entirely inserted into the targeted locus as the vectors are linearized within the homology region. Insertion vectors. Which type of vectors are initially circular but during insertion become linear? Insertion vectors. Which vector has the homology region and it is collinear with target? Replacement vector. Which vector is linearized prior to transfection outside the homology region and then consequently a crossing over occurs? Replacement vector. Introduction of foreign nucleic acids into cells by non-viral methods is called transfection. The whole complement of genes that determine all characteristics of an organism is called genome. Mention the types of proteins present in a genome. Functional and non-expressive DNA proteins. Project in which the whole genome of plant is analyzed using sequence analysis and sequence homology with other plants is called Genome Project. Genome projects has so far been undertaken in the organisms like Chlamydomonas, algae, Arabidopsis thaliana, rice and maize plants. Genome content of an organism is expressed in terms of number of base pairs or in terms of the content of DNA, which is expressed as C value. In recent years, the evolutionary relationship between different plant taxa is assessed using DNA content as well as the similarities and differences in the DNA sequence. Taxa and their relationship are indicated in cladogram. Dash is a group of technologies that has the ability to change an organism's DNA. Genome editing or gene editing, which technology allows genetic material to be added, removed or altered at particular locations in the genome. Genome editing and CRISPR-Cas9. Abbreviate CRISPR-Cas9. Clustered, regularly interspaced, short palindromic repeats, which technique has generated a lot of excitement in the scientific community because it is faster, cheaper, more accurate, and more efficient than other existing genome editing methods. CRISPR-Cas9, in which plant CRISPR demonstrated at first. Rice, who reported in a new study which clearly shows one can re-engineer rice to switch it from a sexual to an asexual mode. Imtiaz Khan and Venkatesan Sundaresan and colleagues named the technique in which copying of genetic information from one strand of the DNA by RNA. Transcription. Dash is a biological process in which RNA molecules inhibit gene expression or translation. RNA interference, in which process neutralizing targeted mRNA molecules takes place. RNA interference. What are the two types of RNAs, two enzymes? Dicer and drosha. In which step dicer and drosha are used? First step of RNA interference. What is risk? RNA induced silencing complex. மேலும் பல தகவல்களை தெரிந்து கொள்வதற்கு பயாலஜி தமிழ்நாடு சேனலை சப்ஸ்கிரைப் பண்ணுங்கள். நன்றி, வணக்கம்.
பலருக்கு சேவை செஞ்ச நீயும் ஒரு ஹீரோடா. ஒரு கைப்பிடி சோறானாலும் ஷேர் செஞ்சு பாருடா.